Hello, everyone, and we'll kick off this symposium now. There is only one ecology. There isn't really a separate terrestrial ecology or a separate marine ecology, and yet too often those disciplines are siloed and different specializations, different foci. And what this symposium is exploring is one way to bring these realms together, and that is through the fluxes between the land and the sea. And that's what we're going to focus on in a range of talks over this symposium.
Yeah, thanks, Yadvinder. I'd like to quickly acknowledge Lisa Wedding as well, who's convened this session with Yadvinder and I, but unfortunately couldn't make it in person. So the idea for this session has come about through a project that we've been running together, looking at the links between seabirds, islands and coral reefs. And we've all really enjoyed it because we've come out of our silos focusing on marine ecosystems or terrestrial ecosystems, and we've worked together and we've learned a huge amount. So we had a workshop earlier in the year, and we thought it would be fun to run a thematic session where we bring together other people tackling these questions in different geographies around the world. And I think you'll agree we've got a great lineup of speakers. We're going from the tropics to the polar regions, and we're going from microbes to the great whales. Our first speaker, our keynote for the session, is Professor Holly Jones from Northern Illinois University. Holly's well known around these halls. She's a regular at the BES. She's a lead editor for Ecological Solutions and Evidence, one of the BES journals. Holly works on restoration ecology across a whole range of ecosystems. She's done some of the seminal work looking at the conservation outcomes of invasive mammal eradications from the world's islands. And she's done some really important work looking at links between islands and marine environments in temperate systems, particularly around New Zealand. So, Holly, the floor's yours.
Well, yeah, thanks, Nick, and thanks everybody for being here. It's an honor to kick off this section, talk a little bit more about the seabird work that we do in the lab, and kind of set the stage for these, land-sea linkages. So, yeah, thanks for the invite. I'm excited to be up here. Yep, doing it. Okay, so if you look at any map, among the first things you'll notice is the boundary lines. And delineations like country, state, county, city, and township can be really useful to our society because they draw lines around governance and laws and social services. But these human-made boundaries are also counterproductive when talking about how to study and manage species and ecosystems. Species and ecosystems don't recognize political boundaries, but laws, scientific funding, opportunities, and so much more really vary across them. And this issue is especially acute when talking about species that span different ecosystems, which is part of what this whole session is about. So these are some of the examples of species that I will call ecosystem connectors. Essentially, they're species that connect various ecosystems with one another in various ways. And before I get to my love, seabirds, I'm going to walk through a couple of examples, sort of set the stage for the rest of the session today. One of the cool things about ecosystem connectors is they're often ecosystem engineers, which we'll get to a little bit later, but it makes their conservation management and research about them pretty important. Okay, so let's get into this frog example. So these frogs live in Panama. These are the red-legged frogs, and they're tree frogs, and they lay their eggs in forest trees that overhang aquatic systems, and their tadpoles then hatch and fall into the water below. And then this is a classic example that many of you have probably heard of. But what's cool about it is it consists of a couple of species and 3 different ecosystems. So Pacific salmon hatch in the river systems where they hatch. They come out to the ocean for a couple of years and then go back to the rivers where they hatched to spawn. And then grizzly bears will come feast on those fish. If anyone has followed Fat Bear Week from Katmai National Park, you will know about this. It's really cool. You should check it out if you haven't. But those bears eat those salmon, and then they go disperse those nutrients throughout the forest. And indeed, a whole lot of nitrogen in spruce trees has been linked to this connection. Whales have their own cross-ecosystem impacts. In a process dubbed the whale pump, whales move from deeper waters up to the surface to breathe, and when they poop, they spread nutrients all over the surface waters, yielding massive phytoplankton blooms and feeding the subsurface food webs. Their movement from their feeding to their breeding grounds has similar impacts. Lastly, both seabirds and pinnipeds, like seals and sea lions, transport the nutrients that they feed on in the ocean back to the places in which they breed. This research by Bockhorst, who we're going to hear from a little bit later, has emphasized how important those nutrients are for shaping animal communities in Antarctic systems. But I don't study pinnipeds, I study seabirds. So for the rest of the talk, we're going to focus on seabirds and their land-sea connections. So true to their name, seabirds spend most of their life on the open ocean, but they have to come ashore to nest, and most often they pick islands to do that on, islands primarily without predators. Seabirds are top predators in the ocean, and they feed on rich marine nutrients through things like fish and squid and a whole host of other things. And then when they come to breed on islands, they deposit that nutrient-rich marine-derived nutrients in the form of guano, shells, carcasses, and a whole host of other things onto the island ecosystems. But they're not done yet. Those nutrients then wash off into nearshore ocean ecosystems, completing what I like to call the circular seabird economy, from ocean to land, back to ocean. So this is a pretty simplistic sort of conceptual figure, and what I want to do today is build out some of the complexity and what we know about these linkages based on previous studies and studies from my own lab. I'm not going to touch on coral ecosystems because, as Nick mentioned, there are a couple of great talks coming up about those coral ecosystems. So coral people, I love you, but I didn't include your stuff because I wanted other people to be able to talk about that. Okay. So let's talk a little bit about the seabirds, about studies on seabirds as connector species. So the classic work in this area was done by Gary Polis and colleagues way back in the early 90s, or mid-90s, I guess, off of studying islands off of the coast of Mexico, both in the Gulf of California and off of Baja, Mexico. And these are desert islands, so a unique ecosystem. And what they found is that on an island that has seabirds, spiders were 8 times as dense inside seabird colonies versus outside seabird colonies. They also looked at lizards and found that islands with seabirds had 4 times more lizards than islands without seabirds. And they did really important work on the impacts of island size and shape, finding that seabirds weren't the only conduits of these nutrients, but also sort of beach wrack that washes up around the shore is important. So they concluded that perimeter to area ratio and size and shape of island was critical for seabird nutrient impacts. All right, so let's build some of this complexity into this model. So they studied biodiversity and found that it varied by seabird influence, and they said that island size and shape really matters. All right, let me see how I'm doing here. Okay, so we're gonna move from these desert ecosystems in Mexico up to the Aleutian Islands. So the Aleutians are that island chain off of Alaska that goes like this, and it's up in the Arctic ecosystem. And essentially the work for the Arctic Aleutians showed that on islands that didn't have seabirds, because invasive foxes had eaten them all to basically nil, the community essentially shifted without that seabird nutrient input from grassland to tundra. And we can all imagine that a huge shift in the vegetation community would have knock-on effects, right, to the rest of the biodiversity, microbes, a whole host of other things included. Okay, so yeah, the nutrients that seabirds provide is really important for those plant communities and shapes them. But also predators matter, right? I was talking about those invasive foxes, and predators are a really big issue for seabirds. They're one of their biggest threats. In fact, their biggest threats on land when they're breeding. Around 90% or more of island archipelagos has had invasive predators invade it, and that's resulted in the seabird circular economy being broken in many ways. And that picture that I just showed you was a good illustration of what happens to communities when those linkages get broken. Okay, the work by Carolyn Curley and colleagues, also in the Aleutians, took us from the sort of the terrestrial environment into the intertidal, and it was really cool work. Essentially what they found was that on islands that did not have invasive predators, seabirds in the intertidal communities were eating herbivorous invertebrates at such high levels that then the intertidal was dominated by macroalgae communities. But when invasive rats invaded and ate those seabirds down to low numbers, those herbivorous snails and other invertebrates increased, and then that resulted in a switch from a macroalgae-dominated community to an invertebrate-dominated community in the intertidal. All right, so let's bring in some intertidal creatures to our wonderful model and account for Carolyn et al.'s work. Okay. So far we've been to the Arctic and to desert island ecosystems. I really wanted to look at what happens in temperate ecosystems for a variety of reasons. So for the rest of this, I'm going to talk about work that stems from my lab. As mentioned earlier, New Zealand is one of the primary places that my lab has done research on these phenomena. And when I was a young graduate student, I was trying to pick where I wanted to study. And I'd worked in the Aleutians for a little bit, and that was amazing. But I also worked in New Zealand for a little bit, and that was great too. I saw the sun a whole lot more in New Zealand, so that was great for its favor. But scientifically, really the reason why I wanted to study in New Zealand was because the technology to get rid of invasive mammals was founded in New Zealand. And so essentially what they had was the longest history of islands that had had predators removed. And so I could look over the longest time possible to look at how these islands recovered, specifically how seabird-driven nutrient dynamics recovered after invasive mammal removal. So I set out to do a natural experiment with islands with different restoration histories as treatments. So I had some islands that had never been invaded before, sort of the restoration goal, if you will. And then islands that were currently invaded, you could consider that a positive control, or the point from which restoration really begins, or recovery begins. And then I had a bunch of islands that had had invasive mammals removed at different times since eradication, so I could sort of track what recovery might look like through time. And I did this in the Hauraki Gulf off of the northern island of New Zealand, highlighted in blue here, but also in the Cook Strait, which is between the North and the South Islands. And I've been saying I was tracking recovery through seabird input into these island food webs, and for those of you that haven't sort of seen that before, I wanted to just walk through a quick primer. Usually when I say isotopes or when I put the delta signal up there, people's eyes start glazing over. I want to promise you it's really not that complex. I'm going to talk you through it really quickly so that you can understand the next graphs that I'm going to show. So essentially, nitrogen on Earth comes in two stable forms, 15 nitrogen and 14 nitrogen. They differ in their mass by just 1 neutron. Marine food chains, because they're longer, are dominated by 15 nitrogen versus terrestrial ecosystems, which are dominated by 14 nitrogen. So when a seabird, as a top predator, goes and feeds in the ocean community on fish that are enriched in 15 nitrogen, and then come back and breed on an island, their guano is enriched then in that 15 nitrogen. So what that means for me as an ecologist, if I go out to any of these islands and I grab some soil, some plants, maybe some spiders, and I measure the ratio of 15 to 14 nitrogen, which is what isotopes do, I can track the influence that seabirds are having in those communities. Which is a long way of saying if you see a high delta 15N, which is the ratio that we use, on these next slides, think lots and lots and lots of fairy prions. Okay, I'm going to set up what these graphs are going to look like before I show you the data. As promised, on the y-axis is delta 15N. It increases as seabird influence increases. On the x-axis, I have time since islands were eradicated, and then I have the islands that have never been invaded will be a line in green, and the islands that were currently invaded will be a line in red. And our hope is that those eradicated islands that will be in the middle of those two lines indicate a positive relationship, so that would indicate recovery towards those never invaded goals. So I took samples of soil and plants and spiders, and the reason why I chose spiders is because in these simplified food webs, they're often one of the top predators on these islands. It's them or lizards because, again, seabirds don't like to breed around other predators. All right, so here's what we found. Essentially, the islands in this study recover from invasion relatively quickly, as shown by those gray error bars overlapping the green lines, essentially, which are never invaded. And, the recovery depends on the variable, but can take anywhere from, 20 or so years on, which may seem like a long time to us, but certainly not for a seabird and certainly not for ecosystems in general. So that's great news. All right, so let's add some delta 15N into this system and move further along. So I was noticing that a lot of the research on seabird impacts and recovery after predator eradication was in terrestrial systems, but I knew this was a circular system where those nutrients wash back into the nearshore community, and I was really excited about figuring out how to study that. And so I recruited a wonderful PhD student who's in the bottom there, who was also interested in marine work. And then I was also co-advising an amazing student who was working in the terrestrial environment. Her name is Steph Burrell. She's looking lovingly at that baby seabird up there. And Steph and Lindsay Rankin, who's the marine student, sort of were studying on the same islands and never really set out to work together, but because they were buddies and were measuring sort of similar things, but one in the terrestrial environment, one in the marine environment, they did decide to eventually team up. So you can see this series of maps here. This work was done in the Mercury Islands of New Zealand, which is again in the Hauraki Gulf, which is the northern part of New Zealand. And then we zoom in as we go. Essentially, we have one island that was a new eradication island, so they had just removed rats the year before the sampling for this. We had another island that had the rats eradicated 30 years ago, so that's Korapuki in the middle there. And then we had two islands that had never been invaded, middle in green. And we had no invaded islands for this study, and that's because the Kiwis are so good at getting rid of invasive predators that it's increasingly hard to get an invaded island as part of your study treatments. Good job, Kiwis. So I'm going to first walk through a couple of things that Lindsay found, and then I'm going to talk about the linking of the marine and terrestrial sampling. But you can see in the maps up here that there are some dots and some transect lines. Essentially, the colors match the watershed coming out. So what we did was we did a digital elevation map, and Lindsay did some fancy stuff in GIS to basically link the terrestrial measurements with the associated transect that was in the marine environment. Okay, so I'm gonna jump into some marine stuff, and then we'll get into like the actual connection type of stuff. So what Lindsay did first was, for these transects, take a quadrat down and look at the macroalgae community composition, so which species were there and in which abundance. And as ecologists know, we love to then make multivariate plots like this one to look at the community differences. So essentially, if you're not familiar with a multivariate plot, that if two dots overlap one another, the macroalgae community composition is the same. And the further the dots get away from one another, the more different community composition is. And the colors in this represent the different island treatments. So you can see that the purple color, the island that had just had the eradication happen, are sort of unique. You know, the macroalgae community composition is pretty unique and sort of in one particular spot. But there's not any difference between the previously eradicated, the one that was 30 years ago, and the never invaded island, indicating essentially that macroalgae communities become more similar to never invaded islands following eradication. And again, this indicates that this sort of seabird circular economy, those linkages are being restored when we get rid of those invasive predators. All right, let's get some macroalgae in there. So next, Lindsay really wanted to study the different processes that might impact how strong the link between land and sea is. And so what she did was look at the different macroalgae species, which are in different colors on this particular graph, by, in this case, depth. And so what the decreasing line means is that there's lower seabird influence in deeper waters. Essentially, in short, macroalgae are more enriched in seabird-derived nutrients in shallower water. Next, she looked at wave action and found that macroalgae communities are more enriched in the seabird-derived nitrogen in calm waters. Essentially, when you get lots of wave action, you get mixing in the water column, and those nutrients aren't as readily absorbed in the macroalgae as they are in calm waters. And then she also looked at runoff, which is sort of how steep the water catchment was. And there were higher marine-derived nutrients in macroalgae communities when the marine community was surrounded by steep cliffs, which is true of a lot of these islands. The last thing she looked at was season. And so in New Zealand, it's pretty temperate. It's not like they have, all 4 seasons like I do where I'm from in the Midwest. Instead, it's either dry or rainy. And in the rainy season, we found that macroalgae marine seabird-derived nutrients were higher, as indicated by the higher bars in these graphs of the same things. Okay, so now we come to sort of the main action where essentially we're connecting the terrestrial variables to the marine variables. So we did this analysis where we looked at the proportion of variation in macroalgae community composition that was explained by the different variables. And in doing so, we directly tested for that connection from land to sea. And we directly tested how strong that connection was on these different islands. And so we had terrestrial variables like slope and aspect and runoff. We had marine variables, which were wave action and depth. We had season, rainy or not. And then we had seabird variables. So this was like seabird density, seabird-derived nitrogen in soils, and whether the island had predators or not, essentially invasion status. And so on these graphs, on the x-axis are the different species of macroalgae that were found on those islands. And then in the y-axis is the proportion of variation that was explained by each category. We also have transect locations. So this is the location of the transects that were done in the marine environment. And essentially this could be considered like a random variable in any model. We know that there might be variation according to where the transect is, but it's not something we particularly cared about in this case. Okay, so what we found on the island that had just had the mammals removed was essentially what you see here. Don't worry about like bar by bar, because you just don't need to worry about that. What you do see is a lot of teal, essentially. Marine variables like wave action and depth are really driving the most of the community composition in these macroalgae species. And if you contrast that with the island that had been, had the predators removed 30 years ago and the islands that had never been invaded, what you see is a shift to a lot of green and a lot of purple. And essentially, what's happening is those terrestrial variables are becoming more and more important, and those seabird variables are becoming more important, indicating a stronger link from the land to the sea. So that was pretty interesting. And kind of the first time where we had a direct statistical link from land to sea in the system. So I was really excited about these results. I'm geeking out on them still. Okay, so we're adding a whole lot of complexity now. Let's add some depth. Let's add some wave action. Let's also add slope and aspect and runoff. And then lastly, let's add season. Okay, so that's sort of all the data I'm going to show you for this talk, and I'm going to round it out by talking a little bit more about what we still need to know. And I guess I've talked, around, 15 or so minutes about what we know about this stuff, and you may think, oh, well, it sounds like it's well studied, right? But what I would say is there's just so much work left to do in these systems. So what I just showed you was just 5 islands in one country that is a temperate ecosystem. I'm really excited to hear the coral talks because we're going to hear more about what happens in these tropical systems with those coral talks. But we still just know relatively little about a whole host of things, and we could use to repeat this work and see how generalizable it is across a whole host of things. And I just want to end with some ideas about what would be cool to look at. And again, if you're a student, maybe just getting started, you want to work on this stuff, please come talk to me or any of the other speakers up here. I'm sure we all have a lot of ideas about what you might get into, but for now, oh, I forgot about burrow density. Yeah, seabird density matters too. But for now, let's think about a couple of things that probably change the strength of these connections. So phenology of seabirds, right, when they're breeding on the islands likely matters. If they're breeding in the rainy season versus the non-rainy season, that likely matters. Their timing of breeding. Body mass is probably important, and albatross has a whole lot more guano than a storm petrel, but no one's actually looked at the differences between the different types of seabirds and how that might impact these systems and linkages. Foraging behavior is another one. So some seabirds will forage really close to where they're breeding and bring food back to their chicks sort of daily, and other seabirds will travel hundreds or thousands of miles to go get food and bring it back to their chicks. And those types of diet items can have different signatures and may influence these dynamics. Nesting modality is another one. So mostly I've worked with burrow nesting seabirds, but some seabirds nest on the surface of islands, others nest in trees, and likely these types of things that we've been studying would vary according to nesting modalities. So it'd be cool to look at. Another thing I've already mentioned is biome, right? So we only have a little information about temperate ecosystems. The coral world is actually getting better studied because there are a lot of folks here that are working on that. So good job, team. But we can use some more studies across different biomes to see how generalizable this stuff is. One other thing I want to mention is climate change. It's kind of the requisite thing to mention. I haven't talked about it yet, but climate change has caused, big die-offs of seabirds in the last few years due to the changing ocean environment, changing their food availability. And so that direct impact can obviously have an influence, but there's also a lot of seabirds that breed on low-lying islands that are likely to be inundated in the near future. And there are conservation translocations happening to new islands that are higher for them, so you can translocate seabirds. Come talk to me if you want to talk more about that. And what would be really cool is to look at those like experiments and ask, how does the seabird circular economy get started as a seabird colony grows? And so anyone who's like really just starting off in their career, if you have a whole career to look at this, it'd be super cool to do. Okay, so just like they connect ecosystems, ecosystem connectors connect across governance boundaries, such as the case for international whaling bans and longline fishing regulations in international waters. These species are often ecosystem engineers, so building the evidence base for the outsized roles in ecosystems can help us both to conserve them, but also the ecosystems they impact. I want to thank the engines for this research, the graduate students who have built the science that you saw here, and I'll also thank you for your time.
Many thanks, Holly. That was really fascinating, and you've set the scene for the session really nicely. We do have a couple of questions, and we have some time. The first one is, do you think that there would be any circumstances where a tipping point is reached so that systems don't recover even after invasive predators are eradicated?
Yeah, this is like the theoretical background for those tipping points is like the theory that I used to inform my dissertation. And I guess the short answer is yes. Yes and, right? So if there are tipping points and there could be a way where we could get seabirds up to high enough densities that would push back over the tipping point. But what I will say is that on these smaller islands, they tend to really respond pretty quickly, and that's nice. But essentially, it's all about the seabirds, and so if they're recovering really slowly, you're gonna get slow or no recovery. And so there's a lot of different seabird restoration techniques that we can use to get them back quicker.
Great, thanks. And the second question, could bird diet affect terrestrial community development? They're thinking about different N to P ratios.
Yeah, absolutely, and someone should look at that. Yeah, I do think it would, and I would love more research on that. Yeah.
Great, thanks very
Thanks.
much. Okay, our second speaker in the session is Sebastian Stiebel. Sebastian has a great love of atolls and has been doing some really important work bringing together atoll ecosystems from across the Indo-Pacific in a range of papers recently. Sebastian's going to talk to us about geosphere-biosphere interactions as a key part of unlocking nature-based solutions.
Yeah, thank you, Nick. So we've heard in the first talk about seabirds as being important connectors as species, but I want to argue that we can also look onto ecosystems as ecosystems that are somewhat the lived connection between land and ocean, and I want to argue that atolls are a fantastic example for such transboundary thinking in a transboundary system. And in this connection between land and ocean, but also in the connection between geosphere and biosphere, then lies are opportunities for unlocking nature-based solutions then in the context of climate change. But before I get started, I just want to make sure that we're all on the same page here. Atolls are these large structures that we find across the tropical Indo-Pacific, these somewhat ring-shaped formations made out of coral reefs, on top of which we find sometimes few, sometimes as many as a couple of hundred islands, and they together then enclose a lagoon in the center. So this is what atolls look like. But the most attention or the most awareness around atolls, and especially in the public perception and in the media, is a very, very specific perspective on atolls. Atolls have somewhat become, or particularly their islands, become the poster boys of climate change. And just like the polar bear is in a way a symbol for melting pole caps, atoll islands have become the symbol of rising sea levels. And of course, atolls are fundamentally threatened by climate change, but this framing has evolved into a very fatalistic view on atolls that sort of, it seems like we've already decided atolls are going to drown within the next decade, and sometimes this goes even as far as some of these articles even feel like we want these islands to drown so that we can prove a point that we want about the urgency of climate change, which some social scientists termed wishful thinking. But even in the scientific literature, when we talk about atolls or when we write about atolls, it's a very reductionist approach down to only the islands, and particularly the smallness of the islands. And in the human context, this then also means that most of the adaptation, in quotation marks, adaptation measures against climate change revolve around measures of migration or translocation. And within this framing of migration or translocation, implicit to this word is that we've already decided we give up the ecosystems. The only thing that we can do is move people away from atolls to other places. But I want to argue that there should be, and there can be, a motivation for place-based management on atolls. Atolls are not just, or the islands are not just the places that humans inhabit. They're also ecosystems in themselves. 40% of them are entirely uninhabited, but they have a biodiversity that is of global importance for conservation. We recently did a study where we looked at the seabirds, which we already heard are fantastic connectors, and found that roughly a quarter of the world's tropical seabirds, or more than 31 million seabirds, are nesting on the Indo-Pacific atolls. That's more seabirds than we find in all of Europe combined. And similarly, many other different species are heavily reliant on atoll islands and atoll ecosystems for their survival, whether it's sea turtles or different species of lowland floras and faunas of the Indo-Pacific. So to protect this unique biodiversity, we sort of asked the question, can we go beyond surrendering atolls or beyond engineering our way out of atolls by building higher and higher seawalls? Can we unlock nature-based solutions to build climate resilience on atolls? And for the rest of the talk, I want to present the paper where we brought together a diverse team of practitioners, researchers in the marine and terrestrial space, and also indigenous scholars, where we argued there is an opportunity for place and nature-based opportunities on atolls. We need to rethink atolls, and key to this rethinking is looking at the land-ocean connection and the connection between geosphere and biosphere. So the first sort of talking point and key aspect is that we have to get away from this framing of atolls being small islands. Atolls are large integrated land-ocean systems. And we try to map out some of the key nutrient and energy flows that we know so far are happening in the atoll. And as you can see from the arrows, of course, the seabirds playing a key role here, but there's many other ways how land and ocean are strongly interconnected in the atoll environment. And we cannot really fully understand processes in the reef or in the lagoon without looking in the terrestrial side. And similarly, looking at the terrestrial space, we would also need to look in the lagoon and other marine processes. But it is not only the energy flow of nutrients or the flow of energy and nutrients between different biological components that we find in the atoll. Another important aspect that is happening in the atoll is also that there's flow of sediments, of geological processes within the atoll system. And to zoom into this model, we sort of argue that the islands of atolls are made up entirely of coral reef sediments. So we can think of the reefs, the coral reefs that surround the atolls, in a way as the sediment factory that generates the sediments, the building blocks out of which these islands are made of. But it's not only the sediment production or the sediment factory in the reef that we need for the islands. It is also, in a way, the conveyor belt that is necessary, the sediment transport from the reef onto the islands. And this is also a finely tuned and delicate system of transportation. And then the islands themselves we can think of as the deposition centers where different biological organisms like plants help to stabilize these islands to grow. And even though all these images, of course, illustrate some sort of a static system, it really means we should view the nature of atoll islands as a nature of growth and a nature of change. So these islands are naturally growing, collecting sediments, but they're also naturally adjusting and changing based on the oceanic boundary conditions in which they exist. And a recently published island change framework by my colleague Paul Kench sort of identified 6 different modes of island change that we find in atoll islands that are completely natural and not necessarily a sign of erosion and climate impact. So islands can be stationary, they can be mobile, just shifting around, they can expand, but they can also contract in size. And from global tracking studies, we find that the majority of atoll islands, over 500 islands that have been tracked over the past decades, the majority of atoll islands are capable of maintaining their land areas or even expanding the land areas. And if we put these 3 talking points together, or these elements together, we sort of argue that we need to reassess what we think is the vulnerability of atoll islands. Atoll islands are not per se vulnerable because they're just a meter in elevation. We argue atoll islands are vulnerable because they slowly lose their natural capacity to grow and dynamically adjust. And this loss in growth capacity of atoll islands comes partly from global change impacts like marine heat waves, like ocean acidification that reduces the capacity of the coral reef sediment factory. But a lot of these impacts are also happening on the local scale, and it is global and local impact together that tip the scales towards atoll persistence. And some of these local impacts are marine-centered, others are entirely island-centered, like deforestation or monoculture farming, which has been linked to elevated rates of island erosion. But some of these impacts also sit exactly in the connection between land and ocean. And one wonderful example of that is the construction of seawalls. We sort of see seawalls being built more and more around atoll islands under the idea of it stops the erosion of atoll islands. But what seawalls are actually doing is they're capping the connection of the sediment transfer from the reef to the islands and limit not the erosion, but the accretion of the islands, the sediment transport. And in these local impacts is a great opportunity. This is not a blame game. We're not saying we're having these local impacts. Rather, we want to argue there's a great opportunity because local impacts can be managed locally. And within that is the opportunity to return the narrative about the future of atolls back to the local communities who can address these local scale impacts. But there's a great deal that we still yet don't know, and this is sort of addressing us as the research community, is to further identify and understand where the biosphere in the atoll ecosystem interacts with the geodynamics, with the geosphere processes that together give rise to this dynamic geo-ecosystem that atoll are. And luckily, we don't have to start from scratch with our research. I really want to emphasize here the power of indigenous and traditional ecological knowledge. Indigenous communities are very much aware of this dynamic nature of atoll islands. And I think a wonderful example is that we in the Western languages, we just call everything an island. But for example, in the Paumotu language, which is spoken in the Tuamotu archipelago in eastern Polynesia, they have many, many different ways for different types of islands. They call some Motu, others Tahuna, others Onetere, Motuone, and encoded in this place naming is how dynamic or how stable an island is, whether it's growing, whether it's shrinking or not. So indigenous knowledge is much more aware of these natural dynamic processes than we currently are, and tapping into this knowledge, I think, is a great opportunity to bring Western science together with indigenous ecological knowledge. So to wrap it up, pathways to place and nature-based opportunities on atolls really revolve around this approach of land-ocean connection. We want to recognize atolls as transboundary systems. They're not just small islands. They're not just ring-shaped coral reefs. Atolls are an integrated unit of marine, freshwater, and terrestrial subsystems. We should also recognize atolls not as static small landforms, but as biogenic structures that have the inherent growth and adjustment capacities. And this growth capacity lies within the connection between land and ocean. And we further want to identify how geosphere and biosphere interact within the atoll geo-ecosystem that can together drive the resilience, or that we can even enhance through ecological restoration. So rethinking atolls, I think, means we want to view atolls as these transboundary biogenic geo-ecosystems that can be stewarded in the future by the local communities rather than being simply surrendered to climate change. Thank you.
Wonderful. Thank you very much, Sebastian. We have a question here on the app. Is there any work being done on how the rates of coral sediment creation changes as coral reefs bleach and acidification increases?
There's work going on, and I think it's important to consider that the reef islands are in a way in an equilibrium with the coral reef sediments. And we found, for example, a study found that after a major bleaching event, when the corals died off, an atoll island actually increased, I think, fourfold in size, because suddenly there was much more sediment available for the islands to build. Over time, this reduced again, but the islands are in a balance with the surrounding reefs, if that answers the question.
Yeah, absolutely. Great. I think we'll open the floor to any questions if anyone's had any thoughts during the course of Sebastian's talk as well. Please put your hands up if you've got a burning question.
I can ask one if it is. This idea of challenging this narrative of atolls being these poster children of climate change. Have you, when you give this talk, say in countries, in atoll countries like the Maldives, how receptive are they to this change of narrative around atolls?
Very receptive. My experience, people don't want to leave. There's a strong connection to the land in most societies, but particularly in these island communities. So most of the communities, they want to stay on their islands and find place-based solutions. And for them it's somewhat frustrating that we already are 3 steps ahead of looking into place-based solutions and just talking about which country can take how many refugees. So there's great reception to this idea of looking into place-based management and fighting for their islands because of the strong relationship to their land. in these communities. Yeah.
Great. Thanks very much, Sebastian. Okay, we're now going to go a bit colder. We're going to move from the tropics to the polar regions. And we've got Steph Bockhorst, who's going to be talking to us about the role of marine-derived nutrients for polar regions.
Okay, thank you, Nick. Yeah, hi, I'm Steph. I'm from Amsterdam. I'd like to take you to the Antarctic to see what the role of nutrients, or marine-derived nutrients in particular, what role they could play in terrestrial biodiversity patterns that we see on land. So to briefly give you a bit of an introduction to Antarctic biology, biodiversity, everything that we see in the Antarctic is shaped by the cold temperatures and the low water availability. So Antarctica supports very few primary producers. Most of them are considered cryptogamic, so mosses, lichens, algae. We've only got 2 vascular plants, so everything is relatively simple, basic. And the only land animals, as it were small invertebrates. I know that we've got penguins, but penguins primarily depend on the marine system for their survival. If we would lock them on land, they would actually die, starve. So real life on Antarctica is basically based on small invertebrates. So this is sort of a simplified vision of a food web. You've got a number of springtails. There's only 10 species in the Antarctic. There's about 36 mites, 250 lichen species, 100 mosses, 2 vascular plants, and a handful of nematodes and tardigrades. And I briefly said most of the biodiversity patterns that we see are shaped by climate, cold temperatures, as you see here. If you go further south, the numbers of lichens, mosses, they actually drop. So it simply gets too cold, it's too dry, they can't survive there. So if you try to grow plants there, even if you give them sufficient amount of nutrients, nitrogen, most of them, they won't be able to benefit from this additional nutrients simply because it's too cold. If you raise the temperature, like we did in this experiment, so we grew them at 2 degrees with ample amounts of nitrogen, they grow a little bit taller, but if you ramp up the temperature, they do much better. So they can really benefit from this additional nitrogen. So temperature is really limiting for the whole ecosystem, and nutrient use is just much more challenging. However, that still raises the question, can these nutrients being brought in by elephant seals or penguins, could they really affect the patterns that we're seeing on land in terrestrial biodiversity? So that means we sampled penguin poo, elephant seal poo, and all sorts of aspects of the terrestrial biodiversity. We did this along the Antarctic Peninsula, so the pointer isn't that good, but almost at the northern tips, those are the most sort of like benign region, the warmest, and we went down south all the way down to Rodwell Research Station with the British Antarctic Survey, the coldest place that we visited. And in each of those locations we collected mosses and lichens, and we extracted microarthropods, so springtails and mites, tardigrades, nematodes, and we quantified their abundance and the number of species. And very obviously became clear that if you sample near a penguin colony or near a location where you've got elephant seals hanging out, you find higher numbers. So the abundance goes up and the number of species goes up. So there's a clear link between having elephant seals and penguins along the peninsula on the diversity and the abundance of the dominant animals that live on land. But how can we really make this connection very strong? Well, you've got to realize that if you ever visit a penguin colony, if you ever go to a zoo, you quite often you smell them. And the thing that you're smelling them, so you smell them before you actually see them, that's ammonia. So if you could quantify the ammonia in the air, which is what we did, if you do that next to a penguin colony, the numbers go quite high up. And if you compare that to a location a little bit further away from the penguin colonies, there's hardly any ammonia in the air. You don't really smell them. So that means there's less nitrogen in the atmosphere, so it doesn't really get deposited on land. So if you then sample all the mosses and lichens that we've collected and actually quantify the nitrogen content, you see a nice gradient. So very close by, high nitrogen content, and if there's no penguins present, the open symbols here, there's hardly any nitrogen in the vegetation. So that sort of suggests that if the quality of your substrate is in low nutrients, it can't support as many springtails and nematodes and tardigrades as you would if you have a higher nitrogen content. There's also a 15N gradient, but I'll come back to that. And thank you for introducing this. That was really helpful. So if you look at the nitrogen content of the vegetation, you can see a very clear link to a strong correlation to the number of species. So the number of species go up, and you also see that the abundance go up. So the largest symbols basically represent a larger number of organisms per gram substrate. That also results in higher microbial activity. So again, we sampled, we quantified microbial respiration in penguin-affected, the blue dots regions, and non-affected, and it is a clear effect of having more nutrients in the system that really promotes microbial activity, and that activity that decreases if you go further away from the penguin colony. And again, there's a very clear correlation, as you would expect from any kind of region, temperate region, that you put more nitrogen in the system, the whole system becomes more productive. But for a polar region, which is mainly driven by temperature and water availability, this is actually quite a novel finding. So there seems to be a microbial component that's responding to this. How does that work to the springtails and mites? Can we actually link them directly to the nitrogen content? And that's where we come back to the stable isotopes. Right? Yeah. So we quantified the delta 15N in the moss vegetation, and we also managed to quantify the delta 15N in the springtails that we found from those samples. And it seems to be a very clear correlation, irrespective of which of the locations we sampled them from. So this is another springtail species that we encountered. Again, there's a pretty strong, almost one-on-one correlation there. And we see the same thing happening with the dominant mites that live in those communities. So those are the types of mites that we're dealing with, the free-living mites. They walk around, they depend on the food that's available in the moss and the lichen communities. They're all positively correlated to this delta 15N. The same happens for the nematodes. There's pretty strong correlation. It fails in the predatory mites. So this is a predatory mite. This is actually eating a springtail at the moment. If you plot that against the vegetation, right, it doesn't work. It doesn't depend on the vegetation for its resources, so you really have to plot this against the prey. But then if you plot the prey against the predators, you get a very nice, almost one-on-one correlation. So that suggests that all that additional nutrients coming in from these penguins and elephant seals sort of affects the whole ecosystem. So the springtails are affected, the mites, the nematodes. We couldn't get enough biomass for the tardigrades, so we don't really know, but the lichens are clearly affected, the mosses are, and all of it basically flows through the whole ecosystem. So one of the nice things about this study is that we sampled along transects. So we could sort of quantify how far the nitrogen from the penguins and elephants, seals basically gets into the air and how far it gets deposited. So there's a sort of like a footprint. You get a number of penguins sitting here, and the nitrogen impact on the vegetation is about 1.5 kilometer. And that same applies for the 15N as well. We get the same kind of trend. Yeah. Luckily also for the sampling that we done this at different penguin colony sizes. So we had penguin colony sizes with about a couple of hundred breeding pairs up to 20,000. And it seems to be a nice clear correlation that the more penguins you have in a colony, the more basically manure gets put on the ground, the more evaporates, and the farther away it actually gets on land. And luckily enough, at the time we were working on this, a colleague of ours was working at Cape Hallett, and they found we're working on an even larger penguin colony. It's unrelated to the study, but their effect basically fell on the same line. So that means we can now sort of start make predictions. If you have a certain penguin colony, we can say, okay, if you've got 20,000 breeding pairs, we would expect that after the impact should be about 1.5 kilometers inland. Now, luckily, there's a lot of penguin biologists working in the Antarctic, and they've mapped where all the penguin colonies are. They've also mapped the colony sizes, so we could link the two. So this is a map, it's a digital map, it's available online. So we made a connection between this map and our correlation that was shown here. So now we could actually start making predictions about where we would find high nitrogen. And we've just shown that high nitrogen is directly linked to high abundance and diversity of springtails and mites. So now we suddenly have a proxy for biodiversity in one of the most inhospitable places on Earth, which is actually very difficult to research. The penguins you can see from the sky, so that actually makes it quite easy to start pinpointing where you would expect a biodiversity hotspot. And as the New York Times said it, actually, if you want to seek like a diversity in Antarctica, you just need to look for the penguin's outhouse. So that's sort of like the main story that I wanted to tell you today. There's a lot of people involved in this work, so I'll lead you to read this. But luckily for us, when this got out, we got a lot of people in the polar community interested on this subject. So we managed to get a special issue out in Polar Biology on the pathways and the impacts of biologically mediated marine and other stored nutrient transfers between polar ecosystems. But it's a great special issue. There's about 12 papers dealing with lichen responses and intraspecific secondary compounds, different types of phosphorus impacts on the communities, or nutrient fluxes between Arctic seabird colonies and to the adjacent coastal marine ecosystems, which we just talked about. So if you're interested in this, please go and have a look at this special issue. Thank you.
Thanks, Steph. I'll be doing some reading. That looks like a great special issue. We have a few questions on the app. First one, how did you sample nitrogen/ammonia in the air? Was there a difference in the lichen communities in their composition as well as abundance dependent on nitrogen? So I think there's two questions there. The first one is about the how did you sample?
Actually, yeah, we had commercial samplers. Basically, it's difficult. It's a couple of years back now. It's basically a simple sampler with some acid on it, and it traps all the ammonia that basically gets blown in the air. We just left it out for a week, and then we collect it back again, and then quantified the amount of ammonia that basically got trapped on the sampler. Yeah. Great.
And then the second question was about changes in community composition as well as abundance.
So we didn't specifically look at the community composition of the mosses and lichens. That's something we should have done, but we didn't actually incorporate it in this study. We were mainly interested in what's actually happening with almost like the easy components that are more likely to respond to the springtails and mites. Yeah.
Yeah. Okay, we have a few more questions if you're willing.
Sure.
Yeah. Have you seen any changes in nutrient impacts as seals or penguins change their diets or behaviour associated with climate change, or would you anticipate any such changes?
I think the biggest changes that we can expect for the Antarctic terrestrial biodiversity is if penguin colonies move away. So if for whatever reason the marine system is changed and they can't find enough food, they have to go somewhere else, find other nesting places, that means that the previous grounds will eventually start losing all the nutrients. And that would lower the abundance and diversity near those penguin colonies. So I think that's the biggest effect that we could expect. Because there's even evidence from historical penguin colonies that have been, left hundreds of years ago, and apparently there is still a nitrogen peak, a 15N peak somewhere sitting in the ecosystem. So I think the legacy will probably reside there for several decades.
Right.
Yeah.
Yeah, interesting. Sometimes as high nitrogen, it becomes toxic. Would you anticipate that to happen in Antarctica?
Absolutely. Once you're right in the middle of a penguin colony, nothing really survives there. Everything just becomes completely toxic. Yeah. But the surrounding environment is so low on nutrients that then the additional nitrogen from the penguins really has a beneficial effect. Yeah.
Right. Great.
People are misquoting my paper in the Netherlands where they're saying, Bockerstael shows that nitrogen is good for biodiversity, but that doesn't apply in the Dutch agricultural ecosystem. Yeah.
Okay. And maybe the last question, how do you know that the ammonium you find in the mosses entered the mosses through the wind and not through organisms?
Not true. Well, there's nothing really to transport them. You've got a penguin colony sitting over here, and a kilometer further away there's mosses growing, and there's no transfer between, there's nothing moving between the penguin colony and and the moss, apart from an occasional scientist. So from that point of view, I don't think there's a logical biological connection that we could think of or could make. Yeah.
Great. Thank you very much.
Okay. Thank you.
Thank you very much. I'll take over chairing the remaining talks of this session. And our next speaker is Jennifer Apu from the University of Seychelles, who has occasionally been sighted chewing leaves in the Indian Ocean. So over to you, Jennifer.
Thank you, Yadvinder. So I know everybody's excited to hear about coral reefs, but just before we get into the impacts of seabirds on coral reefs, I'm going to talk about another ecosystem which we find in the tropical seascape, which is mangroves. And I'm going to show you some of my results on the impacts of seabed-derived nutrients on the mangrove ecosystems. So mangroves are these highly productive coastal forests found at the boundary between land and sea. And mangroves are important habitats because they provide multiple ecosystem services to society. For example, mangroves protect coastlines from erosion. They act as nurseries for numerous marine species. They provide essential resources such as fisheries and timber. And they sequester large amounts of carbon, which contributes substantially to climate change adaptation and mitigation. And the provisioning of these functions and services are reliant on the health and status of mangrove forests, which are increasingly threatened by human activities. So mangroves, they thrive in low nutrient conditions, making them very sensitive to nutrient enrichment. And we know that human-derived nutrient enrichment may negatively impact mangrove forests, which affect not only the goods and services they provide to society, but also the surrounding ecosystems. So we were interested in knowing what happens when mangroves receive nutrient inputs from natural sources, for example, nesting seabirds. So as Holly introduced earlier, seabirds, they feed out in the open ocean, and then they return to their breeding and roosting colonies on land, where they deposit large quantities of seabed guano. And this seabed guano is rich in nutrients such as nitrogen, phosphorus, and other essential micro and macro elements. And the seabed-derived nutrients, they enhance primary productivity around the breeding colonies, but also in niche marine habitats, for example, coral reefs. But we also have instances where seabirds are nesting and roosting in mangrove forests, and we wanted to find out how is this natural nutrient enrichment impacting the mangrove ecosystem. So to do this, we went to Aldabra Atoll in the Seychelles, located in the Western Indian Ocean. So Aldabra is relatively remote and free of anthropogenic influence. Aldabra has huge mangrove forests, around 1,700 hectares of mangroves, which currently represents the largest mangrove forest in the Seychelles. Aldabra also has these huge seabed colonies, for example, over 16,000 breeding pairs of frigatebirds and around 35,000 breeding pairs of red-footed boobies, which are some of the largest colonies in the region. And on Aldabra, these seabirds, they are nesting exclusively in the mangrove forest. So first of all, to start looking at the impacts of these seabed nutrient contributions in the mangroves, we first quantify the amount of nutrients that is being deposited. And we did this by sampling seabed guano and assessing the nitrogen and phosphorus content. We also sampled the mangrove leaves and assessed the nutrient concentration in the leaves, and to determine if there was an uptake of the nutrients by the mangroves themselves. Next, we sampled a mangrove invertebrate food web. Here, I focus mainly on gastropods and crabs, which are some of the most important macroinvertebrates in the mangrove ecosystems, and also their food sources. And again, here, we sampled the nitrogen isotope ratios, like Holly and Steve explained. So because of their high position in the marine food chain, seabirds are enriched in nitrogen-stable isotope ratios, which we then use as a reliable indicator of seabird influence. We also measured the sizes of these gastropods and crabs to see if the enrichment of their food sources had any influence on consumer sizes. And lastly, we wanted to trace the flow of these nutrients to see whether there was an export of seabed-derived nutrients from the mangroves. So here I assessed nutrient concentrations in surface seawater and macroalgae that's in the lagoon. So this is a map of Aldabra. It's a very huge atoll, and all the mangroves are found inside, along the shore of the lagoon. And the seabed distribution is not the same all around the atoll. So we had some sites where there were nesting seabirds and some sites without any nesting seabirds, and then did these comparisons between these two treatment groups. So first of all, using the seabird population size, the breeding metrics, and the nutrient concentrations in seabird guano, we found that the seabirds are actually contributing quite a lot of nutrients to the mangroves on Aldabra. We had a combined total of around 40 tons of nitrogen and phosphorus that is being deposited per year. The nitrogen and phosphorus content was higher in frigatebird guano, but because there's much a larger population of red-footed boobies, they had higher annual nutrient contributions. Next, we looked at the Rhizophora mangroves, which is the most dominant mangroves on Aldabra. So leaves of the Rhizophora mangrove had higher nitrogen stabilized isotope ratios in mangroves where you had nesting seabirds compared to mangroves where you didn't have nesting seabirds, confirming that there was a transfer of seabird-derived nutrients to the mangrove trees. We also looked at the nitrogen and phosphorus content, and here we detected like 39% higher nitrogen levels and 28% higher phosphorus levels in mangrove leaves at seabed sites compared to non-seabed sites, indicating that there was an uptake of these seabed-derived nutrients. Next, we used these nutrient concentrations and looked at the nutrient ratios. Usually, nutrient ratios are used as an indicator of nutrient limitations in mangroves. So here we found lower nitrogen and phosphorus ratios at seabed sites compared to non-seabed sites, indicating that the uptake of the seabed-derived nutrients reduced mangrove nutrient limitations to growth. We also calculated the nutrient resorption efficiency. So this corresponds to the amount of nitrogen and phosphorus that is resorbed from senescing leaves prior to leaf fall. And usually when nutrients become available, the mangroves will, when nutrients become available, the nutrient resorption efficiencies will decrease. However, here we found no differences in both nitrogen and phosphorus nutrient resorption efficiencies at seabed sites compared to non-seabed sites. Next, when we looked at the invertebrate food web, so we had higher nitrogen isotope ratios at seabed sites across all trophic levels, including our basal resources, which was the sediment, organic matter, the epiphytic macroalgae, the mangrove leaves, and also in the consumers. So we had two primary consumers, which was the littorinid gastropods and the sesamid crabs, and then we had one secondary consumer, which was the porcunid crab. So we had a multi-trophic level enrichment of the invertebrate food web at the seabed sites. We had positive relationships between the nitrogen isotope ratios and the nitrogen concentration, especially for the basal food sources, indicating that the enrichment by seabed-derived nutrients and hence the nutrition of these basal food sources. So we had stronger relationships in the sediment, organic matter, and epiphytic macroalgae. We also had positive relationships between the nitrogen ratios and the nitrogen content in the two primary producers, which was the littorinid gastropod and the sesamid crab, but not in the secondary consumer. When we looked at the nitrogen ratios and the sizes of the consumers, we had positive relationships for the two mangrove crabs, the sesamid crab and the porcunid crab. So on average, sesame crabs were 6% larger at seabed sites, and portunate crabs were around 11% larger at seabed sites compared to non-seabed sites, indicating that the enrichment of the food web, of their food sources, were contributing to larger sizes of these mangrove crabs. Next, with regards to the seawater nutrients, so we measured seawater nutrients during both incoming and outgoing tides in the lagoon. And here we had higher nitrate and nitrite concentrations at seabed sites only during outgoing tides, whereas the phosphate concentrations, we had high levels at seabed sites during both the incoming and the outgoing tides. And we coupled that with macroalgae, which we sampled on substrates adjacent to the mangroves, because they have the nutrient signal over longer term. And here again, we had higher nitrogen isotope ratios in the macroalgae at the seabed sites. So overall, we traced the flow of seabed-derived nutrients throughout this mangrove ecosystem, and showing the role of seabeds in maintaining this functional connectivity between oceanic systems and the coastal seascapes, and also their role in improving the nutrient status of these nutrient-limited coastal habitats, for example, mangrove forests. By fertilizing the mangroves where they are nestings, seabeds are likely beneficial to mangrove productivity, and also by increasing the sizes of these mangrove crabs, which are important components of mangrove fisheries worldwide, but are usually regulated by catch size limits. We provide this preliminary indication that seabed-derived nutrients can be beneficial to mangrove crab fisheries productivity. So overall, our results give further support for the restoration of seabed populations, but also their breeding habitats, such as mangrove forests. So I would like to thank all the funders that supported this study and my amazing supervisory team, and thank you for listening.
Thank you, Jennifer. There's a couple of questions online, but if anybody else has questions, feel free to raise a hand and ask directly as well. So question number one, the work and body size increase is so cool. Do you know if those species have higher reproduction or fitness at larger body size, or can you speculate if not?
There are works that have looked at artificial nutrient inputs, and so usually crabs will, when they grow bigger, they will also molt faster and also reproduce more frequently. So there are some work that show that nutrient enrichment contributes to secondary productivity as well. Yeah.
Great. And in the system, are the birds present all year round, or would you expect seasonal effects?
So yeah, so in the tropics, so this comes down to what Holly was saying. There's still a lot to learn about how the different seabeds work. So in the tropics, we have seabeds all year round. And in this system, for example, the frigate beds have this really long breeding cycle, like 18 months. So there's always seabeds in the mangroves, and we also have red-footed boobies that also roost there all year round. So they're constantly present and constantly depositing guano. So of course, my nutrient calculations are only for the nesting seabed population. So it's quite an underestimate because I didn't look at the non-breeding populations as well. So definitely, yeah, they're all year round and just continuously providing these nutrients.
Okay, great. And how far away are the seabird nutrients exported away from the mangroves? How far did you do your collection?
So for my macroalgae samples, the furthest I went was 50 meters out from the mangrove boundary. And then, of course, there is constant flow of with the tide going in and out. So I think they could be exported a little bit further, maybe 100 meters. But we were looking at if we detect any nutrient signals on the coral reefs. So maybe that could be potential follow-up studies.
Okay. And the final one online is one that I was going to ask as well. Actually, do you see any evidence the mangroves are growing better with the additional nutrients? And also, does this influence herbivore activity? Is there any evidence that mangroves are growing more?
So I didn't really look into mangrove growth, but there's been a previous study on Aldabra that's mapped the mangrove extent change over the last 50 years, and they did notice that where the seabed colonies are, we had an increase in mangrove extent, mangrove coverage area in these locations. So potentially that could be one factor that is contributing to extending the mangrove area on Aldabra.
Great. Any question from the audience directly? If not, thank you very much.
Thank you.
Fantastic work. And our next speaker is Hannah Epstein from the University of Essex, talking about connections between coral reef microbes and island nutrient cycling.
Perfect. Hi, everybody. I'm going to be talking with you today. I know the talk says, or the title says, between coral reef microbes and island nutrient cycles. It should probably say the connections between microbes and land-based management. But as we've been talking about in this session, we know that nutrients play a really crucial role in this. So as we've been discussing already, and I'll try to keep this really short, we know that seabirds provide this really important nutrient link between our offshore feeding grounds and island ecosystems. And I'm going to be talking about tropical island ecosystems in this talk. And these nutrient subsidies are beneficial not only for the terrestrial habitat, but also for our marine habitat. And I know, I think Nick is probably going to talk a little bit more about this at the end of the session, but it can provide really important benefits to marine habitats, including things like increasing climate resilience on coral reefs. But of course, seabirds face a big threat, and that is our invasive predators, so our invasive species like rats, which through their predatory behavior can really decimate seabird populations and shut off this shuttling of nutrients. And potentially, this can result in negative impacts in our marine habitats as well. So then if we were able to, if we were able to then eradicate these invasive species, we can then recover our seabird populations and recover those nutrient subsidies entering our nearshore habitats. And I think one of the issues that we kind of face when we're trying to understand these causal links between things like land-based management activities and marine health is that seabirds take years to decades to recover. And this means that, what we see are kind of the recovery of our marine habitats could also take years to decades. And as I think Holly was saying earlier, that's fine for seabird populations, maybe that's fine for coral reefs, but for us, we're not really seeing that in the short term. And for management kind of targets and understanding monitoring of management activities, this is really challenging. So what we really need to find is kind of a early indicator of recovery in a system or in these systems. And I kind of like to think of it as like a reverse canary in the coal mine, right? We're not looking for indicators of stress, but we're looking for indicators of success of our land-based management. Now, I'm a microbial ecologist, so of course I think that the answers might be in the microbes. And despite their really small size, microbes play these really huge roles in our ecosystems and really our global systems, right? Because they play these essential roles in nutrient cycling and recycling. They're kind of biogeochemical potential. And I think what's really special about microbes is that they actually are often the only biological agent that's capable of regenerating elements and inorganic nutrients and organic nutrients into forms that are available for organisms to take up and move up through that food web. So they're kind of these like base of the trophic food web that are really interacting with these nutrients that are entering these systems and having these big kind of flow-on biological impacts through our ecosystems. And you can see kind of the interaction that microbes have around what's happening on land as well. Obviously, the session is all about the land connection. So just as an example of some of our work on the island of Moorea in Polynesia, so this kind of, the psychedelic colors on this map really just represent changes in the community composition of bacteria plankton around the island. And you can see that it's really not homogeneous at all, right? Our bacterial plankton in our water is really kind of all over the place around this map. But you can see that the areas of the biggest change, which are in red, kind of at the top of that island there in those two bays, are really where we have the highest human population, as well as our like large agricultural areas on the island. And so those bacterioplankton are kind of responding in a community, kind of as a community, to these changes in nutrients and changes in land use on the island itself. Now, their ability to kind of respond to environmental change in this kind of rapid way has not really gone unnoticed in the kind of bioindicator field. So it's been suggested plenty of times before that microbes can provide this early indicator of ecosystem stress. So this was a figure that was just published in Marco Terzin's work this year, where they identify that bacteria, archaea, bacteriophages, can be these incredibly early indicators of acute ecosystem stress, right? And this is just because of that ability to respond to those subtle changes in nutrients in the ecosystem. And so I know that this figure is kind of based around stress, but why can't we use these as kind of an indicator for success or recovery of a system when we're kind of looking at these longer term trajectories like we see with seabird recovery? Can we use microbes as this early indicator that things are kind of on track and going forward? So that's kind of the approach that I took with our case study on the atoll of Tetiaroa, which is in Polynesia, just north of the island of Moorea, which I was just showing you, north of the island of Tahiti. And this is a very culturally and biologically important atoll, and importantly has been kind of the subject of invasive species eradications. And I'm going to talk specifically about rat eradication throughout this. Importantly, this atoll is made up of a number of small islets or motu, and at the time that we started studying this island, the whole island had not gone through an entire rat eradication, and that's really important, but some of these atolls kind of represent different rat invasion histories. So kind of what Holly was talking about earlier, kind of we have these variable invasion histories that we can kind of leverage for comparison on a much smaller scale here. So if we zoom into the southeast corner of the atoll, we have Aiae at the top, which has historically been rat-free, is kind of our reference motu. We have Reono down at the bottom, which was eradicated in 2018, which kind of was a trial run for the rest of the atoll, is our recovering motu. And then we have Rimatu, which at the time that we started sampling this, had rats on it, which represents our unrestored motu. So those are kind of our representative management statuses. And we did our due diligence. We looked at the seabird breeding biomass across these three motu, and we can see that it kind of follows this nice kind of linear kind of relationship here. So at our reference motu, we have our highest seabird breeding biomass and our lowest at our unrestored motu. Now, of course, I'm talking about microbes, so we took microbial samples around these motu on the leeward and the windward sides. This is one of my colleagues here collecting some seawater samples, and we did 16S rRNA sequencing for community composition to kind of look at how bacterial communities in the water column vary around these motu and across these different these representative management status islands. So the first thing I'm going to show you, so we have our lovely NMDS plot. We're looking at our community composition of our bacteria. And you can see our reference motu, our recovering motu, and unrestored. And the first thing that you can see is that they're actually quite different. But I think the most obvious kind of pattern that's coming apart is that we're seeing this weird trend in the communities between leeward and windward sides of these motu. So on our reference island over there on the left-hand side, I don't think this thing is going to work very well, but okay. On the reference motu, we see on the leeward sides of the island, which are in orange, they're kind of grouping really, really closely together. All these communities are looking really, really similar and very, very distinct from that windward, the communities on the windward side. And this is likely because this is an island that has high seabirds. It has a lot of nutrients entering that system. And on the leeward side of the island, we're going to have a lot higher residency time of those nutrients. So the microbes on that, on the leeward side are kind of responding to that long-term nutrient input. And it looks really different from the windward because we're getting all of that oceanic influence there, right? So it's a lot more, there's a lot more mixing, there's a lot of differentiation. But interestingly, as we go to our recovering motu and our unrestored, we actually start seeing those communities start to overlap, right? So where they're starting to get closer and closer together until they're completely overlapped on our unrestored motu, which was really, really interesting. So what do these communities actually look like? So I'll walk you through this, but essentially I want you to pay attention to the kind of warm colors and the cool colors. So we have our reference, our recovering and our unrestored motu here. And the actual, these are kind of the top bacterial genera that were present in these communities. And you can see that they're actually differentiating between these warm colors here in our reference motu, which are associated with phytoplankton blooms, whereas our cooler colors, which are more present on our recovering and unrestored motus, are really more kind of oligotrophic, more of our pelagic associated microbes. And so they're actually differentiating here kind of on what we would expect to see with changes in nutrients. So it was really maybe unsurprising, but exciting to have that kind of confirmation. So I'm zooming in really quickly here on two of our genera that kind of have this relationship with seabird breeding biomass here. So this is genus Ecological, where you can see it having this kind of positive relationship with seabird breeding biomass in terms of its relative abundance. And this particular genus is a heterotroph that's involved in nitrate and ammonium assimilation. And so this could be a potential bioindicator of seabird presence if this particular genus is interacting with excess nitrogen species coming into the water from that seabird guano. We also saw a really positive relationship between seabird breeding biomass and Synechococcus, which is one of our picophytoplanktons. It's a cyanobacterium. And these guys are actually very limited in their growth by phosphorus and micronutrients or microelements like iron. And so this particular taxon might also then be interacting with increased phosphorus in the ecosystem from that seabird guano as well. So as I said earlier, Tetiaroa underwent an eradication after some of these data that we collected, and this was in 2022. And so the motu that we have that's unrestored here actually now has no rats on it. It's been eradicated. And we've been following these communities through time. So for my last slide, I'm just going to kind of show you, I plotted very quickly the relative abundances of these two taxa that I was just talking about. So we can kind of see whether we're seeing this initial reaction by those bacteria, or at least in their communities or their relative abundances. So here you have Synechococcus and you have Ecological. The eradication happened in the austral winter of 2022. And for both of these taxa, in the kind of annual cycle previous to the rat eradication, we have really low relative abundances for both of them. And you see maybe a little bit of an increase in summer, just for your normal summer blooms. But then for both of them, 6 months post this eradication, we actually started seeing this increase in relative abundance of these taxa. So it's still really, really low relative abundance, so it's just kind of a beginning trend, but this was really exciting to see and could mean that these could represent really good bioindicators of seabird biomass. So that is all I have for you today. Just want to thank all of my funders and the funders of all of my amazing colleagues on this, and I'm happy to take any questions.
Thank you very much. Okay, so there's a few questions online. I'll pick through them. Do the microbes also reflect the N-isotope values, as we saw?
Yeah, so I didn't. I should have put up all of our isotope values. We do have the isotope values of delta 15N as well, and they are reflecting in some capacity the nitrogen values. But we also have some of the actual water column nutrients, like total nitrogen and our phosphate and our nitrate levels The reason I didn't show those graphs today is just because we don't have them for every single time point that we've been looking at, but we are starting to see trends in diversity of microbes relating to specifically phosphate, which was a really, I mean, probably obvious, but it was a really kind of a nice trend to see. So the microbes do respond to nutrients in the water column as well. It does get a little bit messy, so I'm still working through all of those data, but yeah, hopefully that will come out soon as well.
I'll ask one question. It's a bit unfair because I know this atoll.
Oh, no.
The reference island is quite a small island
It's
compared
quite
to the
a
other
small
two. Is there possibly some sort of island size effect in what you're seeing there?
There absolutely could be. We didn't take that into account for this particular study, although we might be able to add that in because I know that we have, you know, folks that are working not only on kind of island size, but different, you know, I think different types of seabirds are also nesting at different islands on this atoll, and all of the kind of different seascape dynamics. You know, I think it's really nice. We have a lot of folks that are working on this atoll specifically, so we can kind of bring in a lot of the other data to help kind of bulk that out and look at all of the different, you know, what might be driving these differences. Yeah, absolutely.
Great. Thank you very much. Thank you.
Thank you.
Okay, we're going to change body mass, organism mass usually now. I'm going to go to look at whales with Joe Roman from the University of Vermont.
Great. Thanks, everyone. Thanks, Yavinder and Nick, for the invitation. And Lisa, it's really been a great pleasure to visit colleagues and travel around the UK a bit. So, yeah, we're going to switch it up a little bit and talk about, rather than seabirds, talk about whales today. There we go. So there are essentially 4 ecological pathways that we can think about when we're thinking about, and today I'm going to talk mostly about great whales, which include baleen whales, filter-feeding whales, and sperm whales. And that's really a product of their size, not because of their evolutionary history. Most of the work on whale research for decades was their role as predators. What did they eat? How much did they eat? Where did they eat it? What my colleagues and I have been working on for the past dozen or so years is looking more at the nutrient transfer that these whales can provide, as well as the carcasses. So how can that impact community ecosystems, largely in the deep sea? And then there's been work by Jim Estes and some others on the role of whales as prey, especially now that we see whale populations increase. There are more attacks from orcas, and we're starting to learn more things about their behavior and potential trophic cascades. In the ocean, by far, the biggest migration, the biggest movements every day are through diel vertical migration. So the migration of invertebrates or fish that are moving down during the day, perhaps to avoid predation, coming to the surface at night to feed. And in the process, through death, through defecation, there is the sequestration or the pumping of carbon and other nutrients from the surface to the bottom. Approximately 10 gigatons of carbon per year going from surface waters where the photosynthesis occurs to deep waters where organic matter it can be found. When I first learned about this concept back in marine ecology a few years ago, I realized there was something missing. I had been studying North Atlantic right whales for a number of years, and that summer, before I heard about this biological pump, I would see right whales come up to the surface with mud on their bonnets. They were diving a couple of 100 meters beneath the surface, and often when they came to the surface, they come to digest, to rest, to breathe, and also to defecate. And it occurred to me that this might be something worth analyzing. What about these feces? Is it ecologically relevant, or is it, as one colleague called it, a fart in a hurricane? Can we get an idea of what the quantity of this is? You can see in this slide here 2 different ways that whales can interact with the oceans. On the left, you have sperm whales, deep diving whales. They can dive more than a kilometer beneath the surface. They're typically going beneath that mixed layer, feeding at depth and bringing nutrients to the surface, so allochthonous nutrients. Whereas a lot of the baleen whales are feeding closer to the surface within 100 meters or so. They'll dive deeper sometimes. They're doing more like a recycling in that system. And you can see on the upper right, that's a blue whale fecal plume from krill floating at the surface and sperm whales on the lower left. So we've done a couple of different experiments looking at both the fecal plumes and how they compare to other processes. We started out in the Gulf of Maine around 15 years ago, and indeed have found high ammonium concentrations, high phosphorus concentrations, high iron concentrations in these fecal plumes, which are often the limiting nutrients in these systems, and also when we use some right whale fecal filtrate, an increase in productivity in these systems. So on the micro scale, clear indications that these whale fecal plumes can increase productivity and also have a variety of nutrients in particular areas. Since then, there have been studies around the globe looking at these nutrient loads in various cetacean communities. So not only are these changes happening on a local scale, but we can also see them around the world in different places. This movement, this what we call a whale pump, not surprisingly occurs mostly in highly productive areas because that's where the whales are going to feed. So they're helping to recycle or bring some of these nutrients into those areas. But not only are whales moving up and down, moving vertically multiple times a day, they also have the longest migrations of any mammal on the planet. Some whales can travel, gray whales, tens of thousands of kilometers every year. This image here is of humpback whales. They feed off the coast of Alaska and breed in Hawaii and other places. So feeding in these highly productive northern latitude areas, breeding in the lower areas. Baleen whales are capital breeders. What does that mean? They feed for part of the year, especially the females. They're bulking up, storing energy, storing nutrients. And then when they breed, when they have their calves, they're using up that stored energy over time. So they're fasting for a few months a year typically. What's the ecological consequence of this? They're moving nutrients from those high latitudes to low latitudes through urine. The whales aren't feeding mostly in this time, but they are breaking down those stored recesses, the lipids as well as the muscle. And they also release placentas and carcasses and slough skin. This is an image from Hawaii, a heat map there. And what we found is that humpback whales in that system are bringing more nitrogen externally from places like Alaska to the areas where humpbacks breed, more than all the physical processes combined. So they're having a big impact in that area. Also, across the globe, whale populations currently are depleted. It's comparable to what we might see with seabird nutrient movement, except in the case of whales, they're bringing it from thousands of kilometers away, and not every day, but once a year in these enormous pulses. So today's topic is about the movement of nutrients going from these aquatic or marine systems to terrestrial systems. Amazingly, there's more research done on whale carcasses in the deep sea than there are in carcasses stranding on land. I'm not quite sure why that is. It's possible, if any of you have ever seen the exploding whale video, our response often to stranded whales is to try and bury them or to get rid of them, to drag them back at sea. There aren't a lot of studies where you leave the carcass in place and see what the impact is. However, they certainly do attract terrestrial consumers, and I'll show you in a moment. Polar bears, for example, condors and other scavenging birds, as well as flies and other intertidal amphipods. That's an Andean condor in the center there. So the deep sea carcasses, lots of studies of more than 100 endemic species found only on dead whales in the deep sea. We're just starting to understand what this impact might be when whales strand. One study from Kirsten Lydra out of University of Washington looked at a single bowhead whale carcass, estimated to be nutritionally equivalent to about 1,300 ring seals, which is what the polar bear's typical prey is. And polar bears fatten up just like whales, and then they can store that energy, so it's possible the nourishment from just a single dead whale, bowhead carcass, could provide food for polar bears for months to years. That picture on the left is there were 180 polar bears responding to a single stranding in that area. It's possible that polar bear survival benefits from these dead whales, especially during interglacial periods when their hunting is limited. If whales are abundant, they're still going to have the opportunity to get these resources. But we've seen a loss of these marine subsidies over time, and whale populations declined by about 2/3 to up to 80 to 90%. And that shift affected the two species of condors, both the California condor and the Andean condor. Historical for the Andean condor, about 1/3 of its diet came from marine mammals. Now it's about 8% of the diet, and they move inland in response to that. So they've changed their foraging behavior, and that's resulted in a reduction of marine inputs. We've heard a lot today about the measuring how much those impacts are, how much those inputs are. I don't really think we have a sense yet for whales. It really hasn't been measured how it moves through the system, with one exception I'll show in a moment. Similarly, California condor switched from a largely marine mammal diet. This was all looking at isotopes as well, either nitrogen, carbon, in some cases sulfur, in the feathers. So historical feathers, and then compared to contemporary, including some dating back to the Pleistocene. And California condor probably was put at risk when it lost that food supply, but they also started feeding on game animals, which had sometimes had lead bullets in them. So it's part of the reason that lead contamination was a huge issue for California condors, was likely a result of that dietary shift. We do have one hunter that does actually bring these whales onto land, and that's humans, of course. Inuit whalers have been hunting in the Arctic for hundreds of years, for more than a millennia. They arrived in the Somerset Island in about 1200 AD, and that caused increased levels of phosphorus, shifts in diatom community that remain to this day. So these changes from hundreds of years ago, you'll still see them in these freshwater systems there. So that legacy continues, and that's probably the closest we have to starting to get an idea of what the long-term impacts of these nutrient subsidies might be over time. And finally, it's sort of bringing a connection together to what we've heard about today. Whales like seabirds or salmon or sea turtles also, which are moving nutrients there. Whales are bringing it not through their poop, but through their carcasses. Though I didn't mention ambergris, there is one way that occasionally some floats up on land. But these carcasses can provide nutrients that can spread across the continents and through the islands. And with that, I'll take questions and thoughts. Thanks.
Thanks very much. At the moment, we haven't got any questions online, but do feel free to post them or raise your hands. I'll kick off with one while we're waiting. So you showed the dependence of the condor on possibly on whales. This is fascinating. I have two questions from me. One is, that polar bear image was striking. Do you want to speculate on what Arctic systems were like at a time of abundant whales and how they might have been different from the Arctic we know now? And secondly, with the rebound of whales in the last half century, are we seeing condors and other birds shifting back in their behavior?
Yeah, so I mean, the proposal is for us to, and it's just starting to happen now, to change the way we manage strandings. because we do typically treat it as waste, and it's either it's buried, composted, or dragged back at sea. So we haven't seen the response in places like California that we might have, but I have seen in some places there have been shifts in managing those carcasses. As far as polar systems in the past, yeah, I mean, I think importantly I think polar bears as well as foxes and lots of terrestrial species scavenging along the edge, I'm sure would have moved these nutrients around. So, we think of the brown bear relationship to salmon. There could have been very similar pathways historically for polar bears and other species moving these marine nutrients onto land.
Beyond the nutrients, I was wondering whether the trophic structure of the Arctic was different when you had this regular supply of food.
Yeah, it's a really good question. I don't know. The only studies I've seen have been looking at diatoms and moss communities. I haven't seen. Well, so I'll take that back and just quickly. Jim Estey's work on looking on these trophic cascades as a result of commercial whaling in the North Pacific. So when we took the whales out, Jim's work suggests people started hunting pinnipeds. Pinnipeds declined, and then killer orcas started feeding on sea otters, which had an impact on the macroalgae in that system. One potential way that you can see when you're removing these systems that those systems might have changed.
Okay. I'll take one from online quickly. How has the strength of the whale pump changed after the whaling bans? Is there evidence that the whale pump is rebuilding itself?
Oh, you mean I think we haven't seen changes. It's very difficult. I'm very envious of the work that's been discussed today with the Ratty and Birdie Islands. We don't have areas where we can see those changes over time, at least because they were down about 50 years ago. We don't have studies from 50 years ago to now. We can model it and suggest those numbers have gone up, and historically those numbers have increased, but we haven't actually observed empirical changes over time. Yeah.
Thank you very much.
Great, thanks.
And our last speaker is our co-chair, Nick Graham, talking about seabirds and native forests enhance island coral reef energetics.
Thanks, Yudvinda. Great. Well, it's great to present this study to you all. This is the project that we've been running for about 4 years now. You can see there's a big cast of co-authors. The work spans 3 different geographies, so there's authors from multiple countries and institutions, and it's been a real team effort. As we heard in one of the earlier talks by Sebastian, atoll islands are the poster child of sea level rise, with dire predictions about their future. The mainstream media picks this up heavily. It's also in the scientific literature. However, atoll islands are highly dynamic, biogenic systems, and so natural processes can help support their resilience. 3 dominant processes that are really important for atoll islands are reef accretion, which is describing the growth and the building of the reefs themselves, and that's really critical to shoreline protection, to reducing wave energy hitting the islands. Sediment delivery from a whole range of processes on the reef is important for maintaining and building lateral and vertical growth of islands. And then the vegetation is really important in stabilizing the sediments and the soils through their roots. But atoll islands are also highly disturbed and altered ecosystems by humans. One such way that humans have altered these islands is through removal of native vegetation, and predominantly that has been towards coconut plantations. A recent study has shown that in the Pacific, about 60% of forests on atoll islands are coconut plantations. So it's a real dominant problem. Many colleagues call this coconut chaos, the picture on the right there. And then invasive rats are a huge problem, as Holly pointed out in her talk. Invasive mammals, predominantly rats, on around about 90% of the world's island archipelagos. When rats are present, the skies are empty, it's really quiet when you set foot on one of these islands. And that's because the rats are predating on seabird eggs and on seabird chicks. Set foot on an island with no rats, and you can have huge, abundant seabird colonies. The skies are full, it's noisy, and it really stinks from the ammonia in the air. So this project, we've been looking across 3 different geographies. We've been working in the Seychelles, the Chagos Archipelago, and in Tetiaroa in French Polynesia. We've looked at about 28 different islands across those 3 places, and the islands vary in their native vegetation cover. That's depicted in green there, from pretty much 0 native vegetation when it's all coconut plantation, through to about 100% native vegetation where there's been no conversion. And the islands also differ in their rat presence. So we have islands with and without invasive rats. And that, of course, helps drive a gradient in seabird density and biomass. So we've estimated the seabird energy flow that's coming from the open ocean where the seabirds are feeding back onto the islands. Now, the energy flow is a factor of the biomass of different species of seabirds, and you can see in these bars here that their foraging ranges differ somewhat as well. Now, the extent of those bars and the colours differ in part because we've sampled a different number of islands across those 3 different geographies. So if we now look at an island scale, we can show that the seabird energy flow is scaling quite strongly, as one would expect, with seabird nitrogen input to the islands, so the fertilization effect. And these are non-trivial numbers, so as you get up to around 200 kilograms per hectare per year, that's a similar amount of nitrogen that's put into highly intensive agriculture, such as sugarcane farming. So they really are fertilizing these islands. Next, we wanted to look at the energetics of some key components of the ecosystem. So we'll start on the islands, and we've compared on the x-axis here the native forest cover with fine root productivity, so below ground fine root productivity. And you can see that as you increase the amount of native forest cover, there's a very strong relationship with fine root productivity. And interestingly, that interacts with seabird nutrient input, here measured as soil delta 15N. So with lots of seabirds present, you get a steeper slope in that relationship. Interestingly, secondary productivity follows a similar pattern, and these are data that Sebastian's collected. You can see that there's a steeper slope with when there's lots of seabirds on islands and with increasing native forest cover. And this example here is for ground-dwelling arthropods, but we've done a similar thing with lizards and flying arthropods as well. Next, if we look at the corals, now we're looking at seabird nitrogen input on the islands, because it's the seabird nitrogen that's, or nutrients that are getting out into the reef environment. And we've shown in different previous studies how the nutrients get integrated into the coral reef food web. As there's more seabird nitrogen coming off the islands or on those islands, coral productivity increases. Mechanistically, a paper led by Casey Benquit last year showed that the way that the corals are accessing these nutrients is through their algal symbionts. So the algal symbionts of the corals can soak up those nutrients, and then the corals can access those nutrients through the symbionts by actually digesting them. As the seabird signal on the x-axis there increases, coral growth rate also increases. So those seabirds are driving increases in coral growth rate. Now that's really important because what that means is that you get faster recovery rate from disturbances when seabirds are present on islands. So that blue line at the top is the recovery rate of coral cover following a coral bleaching event next to islands with seabirds compared to islands with rats on the bottom. With climate change and coral bleaching becoming so frequent, this rebound potential of live coral cover is quite critical and is quite important to what I'm going to come on to in a moment. And finally, with increasing seabird nitrogen input, we have greater parrotfish energy flow as well. And we've shown in previous studies that herbivores, including parrotfish, can grow 20 to 25% faster when they're living next to islands with seabirds as compared to islands with rats. So next we wanted to look at the role or the influence of these energetics on some key ecosystem processes. So starting on the island again, as you increase fine root productivity, we get it scales with fine root length. Okay, so that's the length of the total length of all the fine roots in a given area in the surface soil. And you can see that we're going up to about 40 kilometers per meter squared there. So really quite incredible the amount of roots that are being put down. Coral productivity is scaling with coral carbonate production. So that's the actual rate at which the corals collectively are building calcium carbonate in the system. And the parrotfish energy flow is scaling with parrotfish sediment production. So parrotfish are feeding on dead reef substrates to access microbial communities, that's what they're feeding on, and they poop out sand. There was a nice study from the Maldives that showed that about 85% of sand-grade sediment is coming from parrotfish. So they're really important for producing that sediment. Next, we moved, we moved to more of a restoration picture. So instead of looking at gradients, we're now looking at islands with rats versus no rats, and islands with below and above a restoration threshold or target for native vegetation. And we're looking at the overall functions that are really important to island resilience to sea level rise. So we'll start with reef accretion potential. So that's the rate at that the whole reef is growing. So that's the coral carbonate production, but also the carbonate production by other organisms, such as calcifying algae. And it's also the infilling of reefs by rubble and sediment that gets bound together and helps build that reef structure. So the darker colours are the next to islands with rats. The lighter colours next to islands with no rats, with lots of seabirds present. And you can see the lines there represent sea level rise projections under a 1.5 degree and a 2 degree warming world. You can see that when there's no rats and we have lots of seabirds, the plus there on that symbol on the left it represents the mean. So on the lagoonal side of these islands, the reef accretion is keeping up with a 2 degree warming sea level rise projection. Whereas if you look at the darker bar, islands with rats are not even close to keeping up with a 1.5 degree warming scenario. On the outside, the ocean side of the atoll, you tend to get much more of a hard reef platform and not so much coral growth on the outsides of these atolls. But even there, we're seeing a 1.5 times increase in reef accretion, and some of that additional coral will be breaking away and helping to contribute to the island itself. Next, we look at the sediment delivery potential. So that's all the organisms that are delivering sediment, not just the parrotfish. The horizontal line in this case is a long-term average sediment delivery that's been estimated for some of these atoll islands in the region. And almost certainly all of those islands that have been studied will have had rats on them because that's the norm, unfortunately. You can see that for the ratty island, the darker blue there on the lagoonal side, the median is sitting right on that average for the long-term mean, whereas for the rat-free island, there's a huge uplift, so 2.3 times increase in sediment delivery. And on the outside of the islands, we're seeing a 2.8 times increase. Now, to put this into context, if you take a hypothetical island that's just over 1 kilometre long and about 150 metres wide, when there's no rats on that island, you're going to be looking at about 150 cubic meters of sediment being delivered to the island in addition compared to an island with rats. So it's a huge difference that this is making. And then finally, the fine root length is really, the function there is the soil binding potential. And you can see that there's a 4.4 times increase in soil binding potential when you're above this native vegetation target as opposed to below it. And the mean there is about 20 kilometers per meter squared of fine roots. So again, a huge influence. And I think what this describes is, this circular seabird economy that Holly described in her keynote, where we're seeing these seabirds bringing nutrients onto the islands, and then the nutrients going off the islands, and then we're seeing feedbacks back to the islands locally as well, and with the vegetation as well, we're seeing these nature-based solutions to support island resilience to sea level rise. So the obvious message here is that eradicating rats and restoring native vegetation can bolster atoll island resilience to sea level rise. There'll be more energy flowing through the system and into it. There'll be increases in core ecosystem processes. Reef accretion will keep up with a 2-degree warming world, reducing wave energy. Substantially more sediment will be delivered to help build and maintain those islands, and there'll be 4 times more fine roots to bind sediments and reduce erosion. Thank you very much.
Thank you very much, Nick. So for a question online, do you think there'd be any circumstances where a tipping point is reached so that systems don't recover even after the invasive predators are eradicated?
So once the invasive predators are eradicated, the timescales over which seabirds return can vary quite substantially. So I think on small, so Tetiaroa we've heard a bit about in French Polynesia, and they're actually seeing quite rapid recolonisation of some of those islands with seabirds. I think because it's a small atoll that has varying rat status already, so there's lots of seabird colonies around to repopulate other islands. So on the scale of 1, 2, 3 years, they're already seeing seabird populations coming back and bringing those nutrients. Some work we did in the Indian Ocean in the scattered islands, it looked more like the timescales over the return of seabirds was along the lines of 10 to 15 years. So much longer timescales. There'll be lots of influences, distance to seabird colonies and the vegetation that's on the islands will greatly influence how long it takes for seabirds to come back and how effectively. Yeah.
And no more questions? Is there anybody in the audience who wants to raise a hand and ask a question? No? In which case we'll just... thank you, Nick. So just a final few comments from me. Just a couple of reflections of 2 things that struck me. And one is, across all our talks, how important we saw the role of animals in ecological function at a planetary scale. And I think too much of our Earth system planetary science has been focused on vegetation and vegetation alone. And this really shows the role that animals play as arteries in the planetary system. And the other thing is the point I made at the start, that too often terrestrial ecology and marine ecology are quite siloed and go to different conferences. And this, I hope the talks today have shown us how fascinating and rewarding it is to actually link terrestrial and marine ecology and have a single ecology. A couple of things to mention. Firstly, for those of you interested in this topic, we noticed there was a community group on terrestrial aquatic linkages. You can see it on that app, and they're suggesting meeting in the Red Lion pub this evening. So you can find the information on the app if you're interested in joining that. Secondly, Nick and I would like to invite the speakers to join us for a collective lunch downstairs now, if you're available. And finally, could we all just thank all of the speakers for a terrific and fascinating symposium. Thank you. brought together very nicely. It's a nice story. So.
Yeah. Cheers. That's a nice story to tell. So.
Yes, yeah.